This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to this exploration of one of nature's most remarkable evolutionary journeys. Today, we'll trace the story of bats, the only mammals to achieve true powered flight. From their mysterious origins over 50 million years ago to the incredible diversity we see today, we'll discover how these creatures transformed from small tree-dwelling ancestors into masters of the night sky. Along the way, we'll uncover the adaptations that allowed them to navigate in complete darkness, hunt with precision, and thrive on every continent except Antarctica. Join us as we unravel the fascinating evolution of bats. Chapter 1. Bats among their mammalian kin. Understanding where bats fit within the great tree of mammalian life has proven to be one of the more persistent puzzles in evolutionary biology. Bats are distinctive creatures, the only mammals capable of true powered flight, with over 1,400 living species distributed across nearly every habitat on Earth except the polar ice caps. Yet determining their closest relatives among other mammals has challenged scientists for decades, with different lines of evidence initially pointing in conflicting directions. Bats belong to a vast assemblage of mammals known as Loriciotheria, named for the ancient supercontinent Laurasia that once dominated the northern hemisphere. This group encompasses an astonishing diversity of forms, from the massive blue whale swimming in ocean depths to the diminutive Etruscan shrew darting through leaf litter, from armored pangolins to fleet-footed horses, from apex predators like lions to the aerial acrobats we call bats. The sheer morphological variety within Loriciotheria makes reconstructing relationships particularly challenging. When you consider that this superorder includes animals as different as a cat and a cow, a whale and a hedgehog, finding the evolutionary threads that connect them requires looking beyond outward appearance to the molecular signatures written in their DNA. Within Loriciotheria, researchers recognize several major lineages. There are the Eulipotyphlans, the core insectivores comprising shrews, moles, and hedgehogs, which represent the most ancient branch, splitting off before the diversification of other groups. There are the carnivorans, containing cats, dogs, bears, seals, and their kin. The perissodactyls comprise the odd-toed ungulates, like horses, tapirs, and rhinoceroses. The setartiodactyls unite the even-toed ungulates, such as cattle, pigs, and deer, with their surprisingly close relatives, the whales and dolphins. The pangolins, or pholidotans, form their own distinct lineage of scale-covered ant and termite specialists. And then there are the bats, the chiropterans, whose position relative to these other groups has required decades of research to resolve. When researchers first examined different types of molecular data, they encountered puzzling conflicts. Nuclear DNA from living species provided strong support for bats belonging to a group called Scrotifera, which unites them with carnivores, pangolins, perissodactyls, and setartiodactyls. Yet mitochondrial DNA from the same species yielded dramatically different results, with bats appearing as an outlying lineage within Loriciotheria, not clearly allied with any particular group. This discordance created initial uncertainty about where bats truly belonged. Recent comprehensive genomic analyses have provided clearer resolution. These studies employed sophisticated phylogenetic methods applied to large sets of protein-coding genes as well as non-coding regions of the genome. The results consistently placed bats within Scrotifera, though the exact arrangement of lineages within this group shows some variation depending on which genes and analytical methods are employed. The current consensus positions bats as sister to Ferunculata, a grouping that includes carnivores, pangolins, and both types of ungulates. Under this framework, bats diverged from the lineage leading to these other mammals deep in evolutionary time, a split whose timing and environmental context will be explored in detail later.
What matters for understanding phylogenetic position is that bats are nested well within Lorasiatheria, sharing a more recent common ancestor with carnivores and ungulates than with any mammals outside this superorder. Some phylogenomic analyses support an alternative arrangement, placing bats within a group called Pegasopherae, specifically clustering them with perissodactyls and carnivores. This hypothesis suggests bats are more closely related to horses and cats than to cows and whales. The difficulty in resolving these relationships definitively reflects the ancient and rapid radiation of Lorasiatherian lineages. When multiple groups diverge from one another in quick succession, the genealogical signal becomes obscured. This phenomenon, known as apolytomy or evolutionary radiation, characterizes the early diversification of Lorasiatheria, creating inherent challenges for phylogenetic reconstruction even with large genomic datasets.
Analysis of transposable elements has provided independent corroboration of bat placement within Scrotophera. Certain transposable element insertions are shared between bats and their Scrotophera relatives, but absent in other Lorasiatherians. These shared insertions serve as molecular markers that trace evolutionary history independently of gene sequences. Chromosome-level organization reveals another dimension of evolutionary relationships. Bats show specific chromosomal rearrangements, inversions, and translocations that occurred after their lineage split from other Scrotophera members. Comparing these rearrangement patterns across species helps reconstruct the branching order within Lorasiatheria and confirms the close relationship between bats and other Scrotophera lineages. The relationship between bats and other Scrotophera members has profound implications for understanding bat evolution. Carnivores, with their specialized predatory adaptations, and ungulates, with their running and grazing specializations, all trace back to a common ancestor shared with bats. The diversity we observe today represents the outcome of tens of millions of years of independent evolution along different trajectories. The dramatic differences between a flying insectivorous bat and a terrestrial carnivorous cat or a grazing horse arose through modification of a shared ancestral genetic toolkit. The precise identity of the common ancestor shared by bats and their closest relatives remains a matter of inference. Given that Eulipotyphlans represent the most ancient lineage within Lorasiatheria, the ancestor of bats and other Scrotophera members was likely a small mammal, probably nocturnal and insectivorous, with a body plan not too different from modern shrews. From this small ancestral form would eventually arise lineages as divergent as bats taking to the air, whales returning to the sea, ungulates evolving to graze open grasslands, and carnivores developing specialized hunting strategies. Comparisons of bat genomes against carnivore and ungulate genomes reveal hundreds of genes showing accelerated evolution specifically in the bat lineage. Many of these genes fall into functional categories related to flight. Skeletal development genes controlling bone growth patterns, metabolic genes enhancing energy production, and cardiovascular genes supporting increased oxygen delivery. These same gene categories show different patterns of selection in carnivores and ungulates, reflecting their distinct adaptations. This pattern of selection is not seen in the lineages leading to carnivores, ungulates, or pangolins. Understanding which mammals are most closely related to bats helps researchers identify which comparisons are most informative for understanding bat biology. If bats are sister to all other Scrotophera, then features unique to bats must have evolved after this deep split. If bats are more closely allied with certain subgroups, then some shared features might reflect common ancestry rather than independent evolution. This phylogenetic framework matters for interpreting both the genomic signatures that distinguish bat lineages and the fossil evidence that traces their history. The Scrotophera placement also constrains hypotheses about what selective pressures drove the divergence. The late Cretaceous and early Paleocene, when these lineages were separating, presented a world of expanding ecological opportunities for small mammals. Perhaps the ancestors of bats began exploiting aerial insects, initiating an evolutionary trajectory toward flight, while their sister lineages specialized for different ecological roles that eventually led to the carnivores, ungulates, and pangolins we see today. The common genomic starting point shared across these lineages would be modified in radically different directions as each group adapted to its particular niche. Having established where bats fit in the mammalian tree through nuclear genomic evidence placing them within Scrotifera, the next question becomes how the bat genome itself reveals the details of their relationships to their closest non-flying Lorisiatherian relatives.
Chapter 2. Genomic Signatures of Divergence. The genome preserves a record of evolutionary change far richer than morphology alone can reveal. When examining how bats diverged from their closest relatives within Lorisiatheria, the comparison with Eulipotyphlans, that ancient lineage of shrews, moles, and hedgehogs, proves particularly informative. These small insectivorous mammals serve as the nearest approximation to the ancestral state from which all Lorisiatherians descended, making genomic differences between bats and Eulipotyphlans especially revealing of what changed as the Chiropteran lineage embarked on its unique evolutionary trajectory. The breakthrough came from sequencing reference-quality genomes for 6 bat species: Rhinolophus pheromequinum, Rusetus aegyptiacus, Philostomus discolor, Myotis myotis, Pipistrellus cooley, and Molossus molossus. These assemblies achieved exceptional completeness, with BUSCO scores ranging from 99.3 to 99.7%, and N50 values indicating highly contiguous sequences suitable for detailed comparative analysis. Researchers compared these bat genomes against genomes from numerous other mammals, deliberately including the Amur hedgehog and Chinese mole shrew as Eulipotyphlan outgroups. The TOGA software, the tool to infer orthologs from genome alignments, identified 4,213 orthologous genes, sequences clearly descended from the same ancestral gene across all species examined. These orthologous sets provided the foundation for tracing which mutations occurred in the bat lineage after it split from Eulipotyphlans.
Gene family dynamics reveal asymmetries in how different lineages responded to evolutionary pressures. When a genome undergoes duplication events, gene families can expand, providing raw material for functional innovation. Conversely, families can contract through gene loss when selection no longer maintains redundant copies. Comparing bats to Eulipotyphlans shows that certain gene families expanded dramatically in the bat lineage, while remaining stable or even contracting in shrews and moles. In one insectivorous bat species, 71 gene families showed significant expansion, compared to just 13 in a fruit bat species, a difference linked to recent transposable element activity. Yet the deeper evolutionary signal appears in families that expanded or contracted specifically in the bat lineage relative to the Eulipotyphlan baseline, reflecting the distinct selective pressures each lineage encountered after divergence. Perhaps the most striking genomic signatures involve DNA repair and damage response pathways. Multiple genes in these systems show evidence of positive selection specifically in bats. The ATM gene, which detects DNA double-strand breaks and coordinates repair, accumulated mutations at rates exceeding neutral drift. Similarly, DNA PKC, RAD50, and QU80, all central to repairing damaged DNA, show selection signatures unique to the bat lineage. The P53 gene, guardian of the genome that triggers cell death when damage exceeds repair capacity, underwent modifications in bats that differ from the Eulipotyphlan version. BRCA2, famous in humans for its role in preventing cancer when functioning properly, also shows bat-specific changes. MDM2, which regulates P53 activity, completes a constellation of DNA repair genes that all bear the marks of positive selection. These changes make functional sense when considered alongside flight's metabolic demands. Powered flight requires extreme energy production, pushing cellular metabolism to generate ATP at rates far exceeding those in terrestrial mammals.
This intense oxidative phosphorylation produces reactive oxygen species as byproducts, molecules that damage DNA. Eulipodophlins, despite their own high metabolic rates, do not face this particular challenge. Shrews scurry frantically through leaf litter, burning energy at impressive rates, but the metabolic intensity of sustained flight surpasses even their frenetic existence. The genomic signatures suggest that as bats evolved flight, strong selection pressure favored variants that could cope with the collateral damage to DNA caused by elevated metabolism. Eulipodophlins maintaining terrestrial lifestyles show no such modifications in these genes.
The immune system tells another dimension of divergence. Bats have lost the entire PYHIN gene family, a set of genes that in other mammals detects microbial DNA and triggers inflammatory responses. The family includes AIM2 and IFI16, both functional in eulipotyphlins and other terrestrial mammals. Their complete absence from bat genomes represents a remarkable deletion. Compensating for this loss, bats expanded their APOBEC3 gene family, which codes for enzymes that mutate viral DNA as a defense mechanism. While eulipotyphlins possess typical mammalian APOBEC3 repertoires, bats show gene duplications that created additional copies with modified functions. The NF-kappa B signaling pathway, central to inflammatory responses in all mammals, also underwent modifications in bats, with changes to regulatory regions that appear to dampen rather than amplify inflammation. These immune changes likely relate to bats' unusual viral tolerance. Bats carry viruses that would sicken or kill other mammals, yet show minimal symptoms themselves. The genomic architecture reflects this tolerance. Fossilized viruses, ancient viral sequences integrated into bat chromosomes from historical infections, reveal a long evolutionary relationship with diverse viral lineages. The pattern of immune gene loss and modification suggests that rather than fighting every infection aggressively, bats evolved to coexist with viruses, tolerating their presence through dampened inflammatory responses, while still maintaining antiviral defenses through expanded APOBEC activity. Eulipotyphlins show no such immune remodeling, retaining the full inflammatory apparatus typical of terrestrial mammals. Limb development genes underwent dramatic modification in the bat lineage. The bone morphogenetic protein pathway, which regulates skeletal growth, shows positive selection signatures across multiple genes. The BMP2 gene provides a specific example. In bat forelimbs, this gene is expressed at higher levels and in broader domains than in terrestrial mammalian forelimbs. This elevated expression drives the accelerated bone growth that produces elongated finger bones, the structural framework for wings. Examination of regulatory regions controlling BMP2 expression revealed bat-specific mutations in enhancer sequences, stretches of DNA that control when and where genes activate. These mutations do not exist in eulipotyphlin genomes. They arose after the split, representing molecular innovations enabling wing evolution. Similar patterns appear in other BMP pathway genes and in genes controlling cartilage development, joint formation, and bone density. Eulipotyphlins maintain the ancestral versions of these developmental programs, suitable for terrestrial quadrupeds with relatively uniform limb proportions. Sensory gene repertoires shifted in opposite directions for olfaction and hearing. Eulipotyphlins rely heavily on smell for navigation and hunting, reflected in their large families of olfactory receptor genes. Shrews possess elaborate whiskers and keen noses. Moles navigate underground using specialized touch receptors. Bats, particularly echolocating lineages, show olfactory gene family contraction. While fruit bats retain substantial olfactory abilities for finding flowers and fruit, many insectivorous bats have reduced their olfactory gene families through pseudogenization, the accumulation of inactivating mutations in now unnecessary genes. The inverse pattern appears in hearing genes. Eulipotyphlins detect sounds in the typical mammalian range, up to perhaps 20 or 30 kilohertz. Their cochlear genes and inner ear development genes maintain ancestral states. Bats evolved ultrasonic hearing capabilities, detecting frequencies above 100 kHz in many species. This required changes to multiple genes. The Preston gene, coding for a motor protein that amplifies sound vibrations in the cochlea, shows particularly strong selection signatures. Genes controlling cochlear bone density and cellular organization also accumulated bat-specific mutations, modifying the inner ear structure to handle ultrasonic frequencies.
Metabolic pathway genes reveal adaptation to flight's energy demands. While both bats and eulepodiflans have high metabolic rates, the specific genes under selection differ between lineages. Bats show positive selection on multiple genes in the oxidative phosphorylation pathway, the biochemical machinery generating cellular ATP through electron transport chains in mitochondria. Specific subunits of complex I, complex 3, complex IV, and ATP synthase all show accelerated evolution in bats compared to eulepodiflans. These changes likely enhance metabolic efficiency, squeezing more ATP from each glucose molecule and each breath of oxygen. Eulepodiflans face different metabolic constraints related to thermoregulation in small-bodied terrestrial mammals and to surviving the feast and famine cycles of ground-dwelling insect hunters. Their metabolic genes evolved under these different selective pressures. MicroRNAs, small regulatory RNA molecules that control gene expression, also show bat-specific variations. Several microRNA families expanded in bats through duplication events, providing enhanced regulatory control over developmental and metabolic pathways. Some of these expanded microRNA families target the same pathways, showing positive selection at the protein coding level, suggesting coordinated evolution of both the genes themselves and their regulatory architecture.
Transposable elements provide independent markers of evolutionary history. These DNA sequences can copy themselves and insert into new genomic locations, leaving dated stamps of when lineages diverged. Bat genomes contain certain transposable element insertions absent in eulepodiflans, and vice versa. Shared insertions present in both lineages must predate their divergence, having occurred in the common ancestor. Lineage-specific insertions occurred after the split. The ratio of shared to unique insertions corroborates divergence timing estimates from other molecular clocks. Particularly active transposable element families in bats contributed to the gene family expansions observed, as insertion near genes can create duplication events or modify regulatory regions. Ultra-conserved elements, DNA sequences that remain nearly identical across vast evolutionary distances due to critical regulatory functions, rarely change. When researchers examined 197 non-exonical ultra-conserved elements across bat and other mammalian genomes, most showed the expected high conservation. However, 3 elements showed marked sequence divergence specifically in vespertilianid bats. These changes are extraordinarily rare, as ultra-conserved elements typically resist mutation across tens of millions of years. Their modification in bats hints at the profound genomic changes required for flight adaptation. Eulepodiflans, examined for the same elements, showed no divergence from the mammalian consensus. Their sequences matched ancestral patterns, indicating stable regulatory architecture.
At the chromosomal level, bats underwent several rearrangements not seen in eulepodiflans. Chromosome painting studies, using fluorescent markers to track chromosomal segment arrangement, reveal inversions and translocations that shuffled gene order along bat chromosomes. These rearrangements did not necessarily alter genes themselves, but brought genes into new regulatory contexts, potentially contributing to bat-specific expression patterns. Eulepodiflan chromosomes retain more ancestral arrangements, providing a baseline for comparison and confirming that the rearrangements occurred in the bat lineage after divergence. The accumulated genomic evidence reveals a lineage that underwent intensive modification across multiple biological systems. DNA repair pathways enhanced to handle metabolic stress. Immune systems remodeled for viral tolerance. Developmental programs reprogrammed for wing construction. Sensory systems rebalanced between olfaction and hearing. Metabolic machinery optimized for flight. These changes, written in the genome sequence, distinguish bats from their eulopodophyllin relatives at the molecular level with the same clarity that wings distinguish them morphologically. The question that genomic signatures cannot answer alone is whether this molecular evidence aligns with or contradicts the interpretations drawn from comparing physical features across species.
Chapter 3. When Bats Diverged.
The question of when bats first emerged as a distinct evolutionary lineage presents a puzzle complicated by conflicting evidence from different sources. The oldest known bat skeletons date to approximately 52 million years ago in the early Eocene epoch. These beautifully preserved specimens already show all the hallmarks of modern bats: elongated finger bones supporting wing membranes, specialized shoulder joints for powered flight, and inner ear structures adapted for echolocation. Yet molecular evidence tells a different story, one that pushes the origin of the bat lineage much deeper into the past. Molecular clocks, those techniques that exploit the steady accumulation of genetic mutations over time, consistently estimate that the chiropteran lineage diverged from its closest mammalian relatives somewhere between 70 and 85 million years ago. This creates a perplexing gap, a dark interval spanning 20 to 35 million years during which protobats must have existed but left no definitive trace in the fossil record. Understanding this discrepancy requires grappling with both the power and the limitations of our methods for peering into deep time.
The molecular clock operates on a deceptively simple principle. DNA sequences accumulate mutations at roughly predictable rates, like sand trickling through an hourglass. By comparing the DNA of living species and counting the differences, then calibrating these counts against known divergent states from the fossil record, researchers can estimate when lineages split from common ancestors. When researchers applied these methods to comprehensive genomic datasets from multiple high-quality bat genomes alongside dozens of other mammalian genomes, they converged on a divergence window in the late Cretaceous period. Molecular analyses placed the split between bats and their sister lineage somewhere between 70 and 90 million years ago. The confidence intervals on these estimates extended several million years in either direction, but did not encompass the 52 million year date of the oldest fossils. This places the origin of the bat lineage firmly in the late Cretaceous period, a world utterly unlike our own. Tyrannosaurus rex still stalked the continents. Triceratops grazed in herds. Marine reptiles swam in warm, shallow seas that covered much of what is now interior North America. Flowering plants had revolutionized terrestrial ecosystems over the preceding tens of millions of years, and insects had diversified in concert with this botanical explosion. Small mammals scurried through the undergrowth, largely nocturnal, largely insectivorous, overshadowed by the dinosaurian megafauna that dominated ecological niches from apex predators to large herbivores. The late Cretaceous world operated under different climatic conditions. Global temperatures were significantly warmer than today, with no permanent ice caps at either pole. Sea levels stood higher, flooding continental interiors and creating extensive shallow marine habitats. The climate was more equable, with less temperature difference between equator and poles. Within this world, somewhere on the landmass of Laurasia, the ancestral population that would give rise to bats began its divergence from the lineage that would eventually produce horses, carnivores, or perhaps both. The divergence itself was not a single event, but rather a gradual process of population subdivision and genetic isolation. Some population of the ancestral mammal, perhaps occupying a particular habitat or geographic region, became reproductively isolated from others. Over generations, mutations accumulated independently in the isolated populations. Natural selection operated on these populations under different pressures, favoring different suites of traits. Eventually, enough genetic and morphological differences accrued that the populations could no longer successfully interbreed even if they came back into contact. At that point, they constituted separate lineages. The end of the Cretaceous brought catastrophe. 66 million years ago, an asteroid roughly 10 kilometers across slammed into what is now the Yucatan Peninsula. The impact released energy equivalent to billions of atomic bombs, triggering wildfires, tsunamis, and a years-long impact winter as debris blocked sunlight. The fossil record shows a sharp extinction event at this boundary, eliminating non-avian dinosaurs, pterosaurs, marine reptiles, and numerous other lineages. The molecular clock estimates placing bat origins in the late Cretaceous mean the bat lineage survived the extinction event. Whatever proto-bats existed at the time, they made it through the catastrophe that ended the age of dinosaurs. The Paleocene epoch that followed provided the ecological space for the bat lineage to evolve its defining adaptations. During the Paleocene, spanning from 66 to 56 million years ago, temperatures gradually warmed after the impact winter dissipated. By the end of the epoch, Earth was entering a prolonged period of exceptional warmth. This culminated in the Paleocene-Eocene thermal maximum, an event that marks the boundary between the Paleocene and Eocene epochs approximately 56 million years ago. During the PETM, global temperatures spiked by 5 to 8 degrees Celsius in just a few thousand years, likely triggered by massive release of carbon into the atmosphere from volcanic activity or destabilization of methane hydrates. This warming event restructured ecosystems globally. The early Eocene that followed saw tropical and subtropical conditions extend to much higher latitudes than today. Palm trees grew in what is now Wyoming. Alligators swam in Arctic waters. Lake ecosystems in the western interior of North America preserved subtropical conditions complete with fish, turtles, crocodiles, and the remarkably diverse insect fauna on which early bats likely fed. The warming trend continued through the early Eocene climatic optimum, which persisted until about 49 million years ago, representing the warmest sustained period of the entire Cenozoic era. It is precisely at this climatic optimum that bats suddenly appear in the fossil record. The oldest complete bat skeletons, dating to 52 million years ago, represent not the origin of bats, but rather a snapshot of an already established and radiating group. They possess fully developed wings and echolocation capabilities, meaning the critical transitions occurred earlier. The absence of fossils documenting this transformation during the dark interval reflects several factors. First, the habitats where this evolution occurred were poor for fossilization. Tropical forests, the likeliest setting for arboreal mammals transitioning to flight, rarely preserve fossils well. The warm, humid conditions promote rapid decay, and the acidic soils dissolve bones before they can mineralize. Second, the populations involved may have been small and geographically restricted. If the transformation occurred in a limited area, the probability of finding fossils decreases dramatically. Third, the transition may have been rapid in geological terms, perhaps occurring over just a few million years, making the window for finding transitional forms quite narrow. The molecular evidence itself offers insight into the pace of change. When researchers examined genes showing evidence of positive selection in the bat lineage, the strength of these selection signatures suggests intense evolutionary pressure over relatively brief periods. Genes under strong positive selection accumulate advantageous mutations faster than the neutral background rate. The pattern observed in bats indicates that once the trajectory toward flight began, it proceeded relatively quickly under powerful selective pressure. Different calibration methods for the molecular clock produce somewhat different estimates, adding uncertainty to the precise divergence date. Some analyses using mitochondrial DNA suggested bats diverged from other mammals much earlier, possibly as far back as 100 million years ago. However, these mitochondrial estimates are now considered less reliable because the mitochondrial genome in bats evolved at an accelerated rate, likely due to the extreme metabolic demands of flight. The faster mutation rate throws off the clock, making divergences appear older than they actually were.
This acceleration reflects the intense oxidative phosphorylation required for powered flight, which generates reactive oxygen species that damage mitochondrial DNA and drive higher mutation rates. Nuclear DNA provides a more reliable foundation, and these estimates cluster in the 70 to 85 million year window. Fossil evidence of bat teeth and jaw fragments from slightly older deposits has been claimed, with some specimens from Portugal and China dated to 55 to 56 million years ago. These fragments extend the bat fossil record back a few million years, but still fall far short of reaching the molecular divergence estimates. Moreover, isolated teeth are difficult to definitively assign to bats versus other small insectivorous mammals without more complete skeletal material. The inner ear structures and wing bones provide the clearest diagnostic features, and these are rarely preserved except in exceptional deposits with fine-grained sediments. Understanding when bats diverged from other mammals matters for interpreting their subsequent evolution. If the split occurred 85 million years ago, the bat lineage had been evolving independently for 33 million years by the time of the earliest fossils. That is an enormous span during which all the characteristic bat features could have evolved. If the split occurred closer to 70 million years ago, the independent evolution spans only 18 million years before the fossils appear. Either way, during this dark interval, the bat lineage evolved from a terrestrial or arboreal mammal into a creature capable of powered flight and ultrasonic echolocation.
The climatic events bracketing this interval, the Cretaceous-Paleocene extinction and the Paleocene-Eocene thermal maximum, bookend a period of mammalian diversification unmatched in Earth's history. The extinction removed the dominant vertebrates and opened ecological opportunities. The subsequent warming created expanded tropical and subtropical habitats where small insectivorous mammals could thrive and diversify. Bats evolved within this window of opportunity, emerging into the fossil record at the climax of Eocene warmth as fully realized aerial insectivores. Their 20 to 35 million year transformation hidden in sediments that have yet to yield their secrets, or perhaps lost forever to the poor preservation of tropical forest environments where the revolution most likely occurred. Chapter 4. Where Bats Arose. The puzzle of where bats first evolved cannot be separated from the question of where their closest mammalian relatives were living when the lineages diverged. The geography of that split should leave traces in both the distribution of early fossils and in patterns of species diversity that persist to the present day. The supercontinent Laurasia, that vast northern landmass encompassing what would become North America, Europe, and Asia, serves as the stage for this evolutionary drama. But pinpointing exactly where within this enormous region the bat lineage emerged requires assembling evidence from multiple sources. The fossil record offers the most direct evidence, limited though it may be. When paleontologists catalog the localities that have yielded early bat fossils, a pattern emerges. The Green River Formation in Wyoming has produced the greatest number of well-preserved early Eocene bat specimens. Recent analysis has established that Icheronecterus gunnelli, found in lower stratigraphic layers, represents the oldest complete bat skeleton yet discovered, dating to approximately 52 million years ago. The formation also yielded Icheronecterus index and the more primitive Onyonycterus fini from slightly younger deposits. The formation preserves an ancient lake system, Fossil Lake, where bats that died or fell into the water were quickly buried in fine sediment that protected their delicate bones from scavengers and decay. The exceptional preservation allows detailed study of wing structure, inner ear anatomy, and other features critical for identifying evolutionary relationships. Europe follows closely behind North America in the early bat fossil record. The Quercy phosphorites of southwestern France have yielded numerous bat specimens from cave deposits slightly younger than those from Wyoming, dating to the Middle Eocene. Germany's Messel oil shale, approximately 48 million years old, contains exquisitely preserved bats, including species with identifiable stomach contents that reveal diet. Africa and Australia have also produced early Eocene bat fossils, though in smaller numbers and generally from slightly younger deposits. The Australian fossil Australonectyrus clarkii from northwestern Queensland dates to approximately 54.5 million years ago, demonstrating how quickly bats spread across vast geographic barriers. The critical observation is that the most primitive bat fossils, those displaying features closest to the hypothetical common ancestor of all bats, come predominantly from North America. Onionycteris, with its claws on all 5 fingers and relatively short wings, represents a more archaic morphology than any contemporaneous European or Asian specimens. Echironectyrus, while clearly a fully developed bat, retains certain skeletal features that link it to the terrestrial mammalian ancestor. These primitive characteristics concentrated in North American fossils suggest the evolutionary innovations that created bats occurred in or near this region. At the time when the bat lineage was diverging from its closest relatives, North America occupied a different position and configuration than today. During the late Cretaceous, the continent was partially flooded by a shallow interior seaway, the Western Interior Seaway, which split the landmass from north to south. The western portion, called Laramidia, consisted of a narrow strip of land running from Alaska to Mexico, bounded by ocean on one side and the seaway on the other. The eastern portion, Appalachia, comprised the area from the present-day Appalachian Mountains eastward. Central America, as we know it, did not yet exist. The Isthmus of Panama would not form until much later, during the Pliocene. By the Paleocene, following the end Cretaceous extinction, the interior seaway had drained, reuniting western and eastern North America. The Rocky Mountains were actively rising, created by tectonic forces as oceanic crust subducted beneath the western edge of the continent. The climate was warm and wet, supporting extensive forests that stretched across much of the continent. The mammalian fauna recovering and radiating after the extinction included early representatives of the groups that would become modern orders. Somewhere in this Paleocene North American landscape, the protobat lineage was evolving its distinctive adaptations. The early fossil record of other Loraciotherian lineages provides geographic context. Carnivores, perissodactyls, and cetatardiodactyls all show early representatives in North America during the Paleocene, supporting the hypothesis that this region served as a major center of mammalian evolution when bats were diverging.
However, North America did not exist in isolation. Throughout the late Cretaceous and Paleocene, intermittent land connections linked North America to Europe across the North Atlantic. These connections, sometimes called the Thulean route after the mythical northern land, allowed terrestrial fauna to disperse between the continents. Fossil evidence shows that many mammalian lineages appear on both sides of the Atlantic during this time, sometimes with near-simultaneous first appearances that suggest active migration. The ancestral populations from which bats emerged could have ranged across both continents, with the actual divergence from sister lineages occurring anywhere within this connected Laurasian region. Asia, the third major component of Laurasia, presents a more complicated picture. Connections between Asia and North America via Beringia, the land bridge that periodically emerged across what is now the Bering Strait, existed during the Paleocene. Connections between Asia and Europe were more variable, depending on sea levels and tectonic movements. Early mammalian fossils from Asia are less well known than those from North America and Europe, partly due to less extensive fossil prospecting and partly due to differences in depositional environments that affected preservation. The possibility that bats originated in Asia cannot be definitively ruled out, though the fossil evidence points more strongly to North America.
The question of why the bat lineage diverged where it did relates to ecological opportunity and the distribution of resources. Flying insects, the presumed food source for early bats, were globally distributed during the late Cretaceous and Paleocene. Night-flying moths, beetles, and other insects existed on all continents. However, the diversity and abundance of these insects varied with climate and vegetation. The warm, forested environments of Paleocene North America would have supported rich insect faunas, providing ample opportunity for predators capable of catching prey in flight. The concurrent presence of related Lauraceatherian lineages in the same regions where early bats likely evolved raises questions about competitive interactions and niche partitioning. If bats evolved from small terrestrial or arboreal insectivores sharing habitats with the ancestors of other Lauraceatherian orders, what allowed the bat lineage to persist and diverge rather than being outcompetent? The answer likely lies in the nocturnal aerial niche. By evolving flight and specializing on night-flying insects, protobats exploited resources that terrestrial carnivores could not effectively access. The ancestors of modern carnivores were pursuing different prey, perhaps small vertebrates or larger insects, using terrestrial hunting strategies. The explosive appearance of bats across multiple continents by the early Eocene, within just a few million years of the oldest definitive fossils, demonstrates their dispersal capability once powered flight had evolved. From a presumed center of origin in North America, or possibly Central America, bats spread eastward to Europe, westward across Beringia to Asia, and eventually to Africa and Australia. The favorable climatic conditions of the early Eocene would have facilitated bat dispersal by providing continuous suitable habitat across vast geographic ranges. Europe received bats from North America across the North Atlantic land connections, which were intermittently available during the early Eocene. The fossil bats from France and Germany show morphologies distinct from their North American contemporaries, suggesting that differentiation and speciation had already occurred by the time these European populations became established. Some European fossil bats show features that align them more closely with certain modern families, indicating rapid diversification was underway. Asia's connection to North America via Beringia provided a dispersal route for bats to reach the Old World. Fossil evidence from China, though limited to isolated teeth and jaw fragments for the earliest periods, confirms bat presence in Asia by the early Eocene. The subsequent spread into the Indian subcontinent, which was still an island continent drifting northward toward its collision with Asia, introduced bats to what would become another major center of diversity. Fossils from India, dating to approximately 54 million years ago, represent some of the oldest bat remains outside North America, suggesting relatively rapid dispersal eastward once the lineage achieved flight. Africa's isolation from other continents during the late Cretaceous and early Paleocene means that bats almost certainly arrived there via dispersal, rather than being present when the continent separated from South America and Antarctica in the breakup of Gondwana. The early Eocene bat fossils from northern Africa suggest entry from Europe or possibly from Asia, taking advantage of the geographic proximity between continents in this region. The subsequent evolution of African bat faunas, including the origin of certain families found only on that continent, reflects the combination of immigration from Laurasia and in situ diversification. Australia represents the most puzzling case. The continent had been isolated from other landmasses for tens of millions of years by the time bats arrived. For bats to reach Australia required crossing significant water barriers, likely via island hopping through what is now Indonesia and New Guinea. That this crossing occurred so early, within just a few million years of the first bat fossils elsewhere, demonstrates the remarkable dispersal ability that flight provided. Modern bats regularly cross oceanic barriers and colonize remote islands, and the earliest bats evidently possessed similar capabilities.
The rapid global dispersal of bats during the Eocene contrasts sharply with the more limited ranges of their closest mammalian relatives. Carnivores, ungulates, and pangolins spread across continents too, but their distributions were constrained by the need for continuous land connections. Oceanic barriers that bats could cross with relative ease remained impassable to terrestrial mammals until much later geological periods, when land bridges or rafting opportunities arose. This difference in dispersal capability meant that bats achieved a global distribution far more quickly than their sister lineages, even though all these groups were radiating from Laurasian origins at roughly the same time. The biogeographic history encoded in modern bat diversity patterns still reflects this ancient Laurasian origin and Eocene dispersal. While bats are now found on every continent except Antarctica, with maximum diversity in the tropics, the relationships among bat families and superfamilies show a clear geographic structure. The molecular phylogenies reveal that 3 of the 4 major microchiropteran lineages originated in Laurasia, consistent with a North American or Central American origin point. The 4th lineage shows evidence of Gondwanan origins, but this likely reflects secondary dispersal into the southern hemisphere followed by diversification rather than an independent origin of bats in Gondwana. Where bats split from their closest mammalian relatives appears to be inseparably linked with the Laurasian region where all these lineages were diversifying during the Paleocene. The concentration of early fossils in North America, the phylogenetic relationships suggesting Laurasian origins for most bat lineages, and the presence of related mammalian orders in the same region during the same time period all point toward this conclusion. Yet the precise location within North America or Central America, the specific habitats and ecological circumstances that fostered the evolution of flight, remain subjects for speculation until fossils from the critical Paleocene period are discovered.
Chapter 5. When Morphology Misled. The most influential mistake in bat systematics came not from careless observation, but from careful study of neural pathways in fruit bat brains. In the late 1980s, a researcher examining the visual system of megachiropterans discovered that the route connecting the retina to the midbrain followed a pattern seen in primates but not in other mammals. This advanced visual pathway, combined with other anatomical observations, led to a startling proposal. Large fruit bats shared a closer evolutionary relationship with primates and flying lemurs than with other bats. The flying primate hypothesis, as it became known, suggested that powered flight had evolved twice independently within mammals, once in megabats and once in the echolocating microbats. If correct, bats were not a natural evolutionary group at all, but an artificial assemblage of distantly related lineages that happened to look similar because both had adapted to aerial life. The hypothesis rested on more than neural anatomy alone. Immunological studies comparing blood proteins across species provided apparent corroboration. Researchers at the University of Heidelberg measured how antibodies raised against proteins from one species reacted with proteins from others. Greater similarity in antibody reactions supposedly indicated closer evolutionary relationships. When they compared blood serum proteins from megabats with those from primates, they found similarities suggesting taxonomic affinity. Microbats showed different protein patterns. The immunological data seemed to support what the brain anatomy had suggested. Megabats belonged with primates, not with other bats. For systematists, the implications were profound. All the elaborate wing structure, the specialized shoulder anatomy, the modified cardiovascular system, every distinctive feature that made bats recognizable would have had to evolve twice from scratch. The hypothesis demanded acceptance of extraordinary convergence. Yet the visual pathway evidence seemed compelling enough to warrant serious consideration, and the immunological data appeared to provide independent molecular support. Opposition came swiftly from other morphologists. The structure of the wing itself presented the central problem. Bat wings, whether in megabats or microbats, share an identical fundamental architecture. The humerus connects to an elongated radius, the ulna reduces, and the metacarpals and phalanges of digits 2 through 5 extend to extraordinary lengths, supporting a membrane of skin. The thumb remains short and clawed. This precise pattern, replicated in every detail across all bat species, seemed implausible as an independent evolutionary invention. Bird wings evolved from the forelimb using a completely different developmental program, incorporating different bones and forming feathers rather than skin membranes. The pterosaur wing used yet another solution, with a single elongated fourth digit supporting most of the membrane. That two mammalian lineages would independently evolve the same 5-digit solution, down to which fingers elongate and which remain short, strained credulity. Other skeletal features accumulated as evidence for bat unity. All bats possess a culcar, a unique cartilaginous spur projecting from the ankle that supports the membrane between the hind limbs. This structure appears in no other mammal. All bats have modified shoulder joints allowing extreme mobility during flight, with the scapula positioned laterally on the rib cage rather than dorsally as in terrestrial mammals. All bats have distinctive rib cages with flattened ribs that reduce weight while maintaining structural support for the flight muscles that attach to them. The sternum is enlarged and keeled to anchor the powerful pectoralis muscles. Could two independent lineages evolve not just flight, but the same detailed biomechanical solutions? The echolocation debate intensified the controversy. Microbats produce ultrasonic calls and listen for echoes to navigate and hunt in darkness. Most megabats lack this capability, relying on vision instead. If megabats had split from microbats deep in evolutionary time, their lack of echolocation posed no puzzle. They simply never evolved it. But if megabats and microbats were sister groups sharing a common ancestor more recently than either shared with other mammals, then the distribution of echolocation required explanation. Either it evolved in the common ancestor and megabats lost it, or it evolved independently in microbats after divergence. Loss of such a complex sensory system seemed improbable unless vision provided an adequate substitute, which it evidently did for fruit-eating bats. But the molecular evidence would eventually show that echolocation did evolve in the common ancestor, and certain megabat lineages did lose it, while others retained rudimentary capabilities.
The resolution came from DNA sequencing. As molecular biology techniques advanced in the early 1990s, researchers began comparing gene sequences from bats and other mammals. Every molecular study found strong support for bat monophyly, the hypothesis that all bats descended from a single common ancestor. Megabats and microbats consistently grouped together, not with primates or any other mammalian order. The epsilon globin gene provided particularly compelling evidence. Globin genes code for proteins that bind oxygen in blood, and they evolve at rates useful for reconstructing ancient relationships. When researchers sequenced the epsilon globin gene from megabats, microbats, primates, and other mammals, the bat sequences clustered together, while primate sequences formed a separate group. The statistical probability that this pattern could occur by chance if megabats were actually related to primates approached zero. Other genes told the same story. The gene encoding interphotoreceptor retinoid binding protein involved in visual pigment recycling showed bat monophyly, despite its role in the visual system that had inspired the flying primate hypothesis. BRCA1, a gene involved in DNA repair, placed megabats with microbats. The FOXP2 gene, associated with vocalization and motor control, gave identical results. Crestin, the motor protein gene critical for high-frequency hearing, showed that the molecular machinery for ultrasonic detection existed in the common ancestor of all bats, even though megabats had reduced or lost the echolocation behavior itself.
The molecular consensus became overwhelming. Study after study, gene after gene, analytical method after analytical method, all converged on the same conclusion. Bats formed a single evolutionary lineage. Molecular phylogenies placed this lineage within Scrotifera as relatives of carnivores and ungulates, definitively rejecting any primate relationship. What had gone wrong with the morphological analysis? The visual pathway that inspired the flying primate hypothesis turned out to be more evolutionarily labile than initially appreciated. Later studies found that the supposedly primate-specific pattern occurred in some other mammalian lineages as well.
Its presence in megabats likely reflected either retention of an ancestral feature that other mammals had lost or independent evolution of the same pathway. Megabats rely heavily on color vision for finding ripe fruit in dense tropical forests. The neural architecture supporting this visual ecology need not indicate phylogenetic relationship with primates, who developed similar pathways for their own ecological reasons. The immunological studies suffered from technical limitations that plagued many early protein comparison methods. Antibody cross-reactivity depends on protein surface chemistry, which can be influenced by factors other than evolutionary relatedness. Proteins under similar selective pressures in different lineages can converge on similar structures, producing antibody reactions that mislead about phylogenetic position. The method lacks the precision and objectivity of DNA sequencing, where the four-letter genetic code provides unambiguous information about similarity and difference.
The deeper question is why morphology misled so badly in this particular case. The answer lies in the overwhelming influence of flight on body form. Powered flight imposes extreme selective pressures on anatomy. Every aspect of the skeleton, musculature, and physiology must be optimized for the demands of staying aloft and maneuvering in three-dimensional space. These pressures drive convergent evolution, causing distantly related flying organisms to evolve similar solutions. They also cause rapid evolutionary change in features that systematicists typically rely on for reconstructing relationships. Birds, bats, and pterosaurs all independently evolved wings, but each used different developmental programs and anatomical building blocks. The convergence operates at the level of function, not implementation. Within bats, however, the convergence was so complete, the selective pressures so uniformly demanding, that the morphological transformations required for flight obscured the phylogenetic signal that would otherwise reveal relationships. The elaborate specializations evolved once, in the common ancestor of all living bats, after the chiropteran lineage diverged from other scrotatifera. But those specializations so dominated the bat body plan that anatomists looking for evolutionary relationships found themselves misled by features shaped more by physics and aerodynamics than by ancestry. The controversy surrounding bat origins demonstrates how different types of evidence can conflict and why molecular data ultimately prove decisive. Morphology excels at revealing adaptations, at showing how organisms solve ecological problems, at documenting the transformations that evolution produces. But when adaptive pressures become extreme, when convergence is pervasive, when the traits that dominate our observations are the ones most strongly shaped by selection for a particular lifestyle, morphology can obscure rather than illuminate genealogy. The resolution required looking beyond what we could see in skeletons and anatomy to what was written in the genome itself, where the historical record of common ancestry remained legible despite the dramatic morphological transformation that flight imposed on the bat lineage. The evolutionary story of bats reveals how multiple lines of evidence converge to illuminate deep time. From their placement within Laracitheria alongside carnivores and ungulates, to the genomic signatures of DNA repair and immune system remodeling that enabled flight's metabolic demands, to the mysterious 20-million-year gap between molecular divergence estimates and fossil appearance, bats challenge us to reconcile what genomes reveal with what stones preserve. Their rapid global dispersal from probable North American origins demonstrates flight's transformative power for mammalian evolution. Perhaps most remarkably, the resolution of the flying primate controversy underscores a fundamental lesson. When selective pressures are extreme, morphology can deceive. The very adaptations that make bats extraordinary, their wings, their metabolism, their sensory systems, so thoroughly reshaped their bodies that only molecular data could pierce through convergence to reveal true relationships. Bats remind us that evolution's deepest truths are often written not in bones, but in base pairs. Thank you for listening.